It's odd for a week that only has 4 days that today already seems like Friday to me. Does it seem like that to you guys too? It's like, that's too weird. So, I've already had several of you come visit me about exams, so I'm happy to see that, and had some good conversations. And so please do schedule a meeting with me, and we can, or come by. You can just come by. I am going to be make myself available. My schedule is pretty crowded. If you looked at my online thing, you can see. But I will make myself available tonight after the class. So if you have things ready to go, to meet with me, you need to have, of course, everything filled out and so forth. So don't just come and sit and talk. But I want to see what you've got in terms of what you put for answers on your exam. And I will do that again on Friday as well. So I'll probably be more available on Friday than I will tonight. So tonight I'll be available for a little bit. Friday I'll make myself a little bit more available than otherwise. These are due no later than Wednesday of next week at 5 p.m. What was I going to say? I will get them back to you by Friday so that you'll have something to study for the final exam. Okay? So that's the plan. Okay? Yes? By then. That's correct. That's correct. So this is it's not just a matter of turning things in. You have to meet with me if you're expecting to gain anything from this. Okay. And I'm doing that not to be mean, but I think it's important that we talk. I find that I hopefully can help you a little bit as I'm talking to you, but part of the problem I think is just distance, and we're not talking. So I want to encourage that. Okay. Okay. All right. I want to finish up a couple things on this topic of electron transport and oxidative phosphorylation. They're actually peripherally related to the problem, or I shouldn't say the problem, but to the phenomenon. It's not really a problem as long as it's working. It's when it's not working that we have problems. So one of the considerations about the electron transport and oxidative phosphorylation is actually a sort of a problem. And the problem as such is that NADH and FADH2 do not cross the inner mitochondrial membrane. They don't cross. Okay? They're too big. They won't go across there. So in order to get electrons in from the cytoplasm, the cell has to use a sort of a trick to get them in. They use molecules that carry electrons that can be transported across the membrane. And then when they get in, they give up their electrons that can then be used in the electron transport system. So there are two systems that are commonly described. One is more commonly found in insects, and it's the glycerol phosphate shuttle that I'll show you here. So how does it work? The way that the shuttle works is actually pretty simple. It turns out that inner mitochondrial membrane has a transport protein that will move glycerol phosphate from the cytoplasm into the matrix. Okay? Now, NADH and NADPH will not go across there by themselves, and there's no transport system for them. So to get electrons from the cytoplasm, from, in this case, NADH, the way that they're gotten in is that the electrons are donated to dihydroxyacetone phosphate. Where have you seen that guy? What pathway? Okay, so there's a point of extra credit. Yes. Okay, so DHAP is a molecule from glycolysis. When you give 2 electrons and 2 protons to it, you make glycerol phosphate. Glycerol phosphate can be moved in via a shuttle protein, okay? And when it gets in there, then the reverse reaction happens, okay? Or at least to some extent the reverse reaction happens. The electrons are donated to an electron carrier, but in this case they're donated to FAD to make FADH2, and that produces DHAP, which then gets transported back out. Okay? So the electrons have been carried by a shuttle molecule. In this case, the shuttle molecule is glycerol phosphate. Glycerol phosphate is the shuttle molecule carrying the electrons. They get transferred to FAD to make FADH2, and dihydroxyacetone phosphate gets moved back out. So we see basically a cycle, right? And what we're doing in this cycle is we're converting electrons from NADH to FADH2. Now, there's a problem with that. The problem is that if you recall, electrons from FADH2 get transferred to which complex? Complex 2. And the electrons from NADH get transferred to which complex? Complex 1. So what does complex 1 do that complex 2 does not do? Pumps protons. So NADH electrons essentially have more potential energy than FADH2 electrons do. Because electrons from NADH go through complex 1, 3, and 4, right? There's other ones also, obviously, but 1, 3, and 4 all pump protons. But FADH2 brings electrons into complex 2, and they only go through 3 and 4. So there's not as many protons pumped for each FADH2 as there is for each NADH. So this turns out to be an inefficient process. Why do cells use it? Well, insects use it in their muscle, and they use it because it's very quick. It's a very quick way to get electrons in, and it's not the most efficient, but I'll wager you that the fastest hot rod on the road is not the same as a Prius in terms of efficiency, right? It gets speed, but it doesn't get efficiency. This thing gets speed, it doesn't get efficiency. Tessa? It's primarily an insect muscle. We can do this to some extent as well, but it's primarily a consideration in insect muscle. And it's one of the reasons why insects are more susceptible to rotenone. Okay? So this is a pretty important thing for insects. Insects need to have very fast twitch muscle, right? Okay? Now, the primary system that we use is actually not this system, but it's a more complicated system called the Maleate-aspartate shuttle. Now, this looks a lot more complicated. I'm going to simplify it a lot for you. Okay? The Maleate-aspartate shuttle involves several molecules, but the molecule doing the shuttling is malate. Malate is carrying the electrons from the cytoplasm into the matrix because there's a transport protein that will move malate in. So malate is the shuttle molecule, and the last one, glycerol phosphate, was the shuttle molecule. Okay? Well, how do we get the electrons on there? We start with, there's our friend oxaloacetate. We add electrons to oxaloacetate. It's the reverse reaction of the citric acid cycle to make malate. Electrons in, and then it goes back to oxaloacetate. Everybody follow? Oxaloacetate gets electrons to become malate. Malate goes in, gives electrons back to make oxaloacetate. And look what happens here. The electrons start with NADH, and they end up with NADH. This system is slower, but it's much more efficient because we don't lose any of those protons that would be pumped, because this guy is going to go to complex 1 just like this guy would go to complex 1 if it could. Make sense? So, yeah. Question?
Yeah. So,
what's all this crap, right?
if it goes to oxaloacetate in the matrix, then how does it get out if it can't cross the membrane?
Okay. So, how does oxaloacetate get out? Okay. So, the rest of this, or at least the answer to what you're asking, and the rest of this is balancing the equation. Oxaloacetate doesn't make it across, which is why all this other stuff's here. What can make it across is aspartic acid. Now, I'm going to show you this. I'm not going to hold you responsible for this up here. This is just balancing equations, and in my opinion, balancing equations isn't the most important thing that's here. The most important thing is what's doing the shuttling. Oxaloacetate gets converted to aspartic acid by a transamination, meaning that an oxygen is swapped for an amine. That makes aspartic acid. Aspartic acid can get popped out here, we complete the equation. But then the question is, well, where in the hell did the amine come from? That's where all this business is. The amine comes from glutamic acid, leaves alpha-ketoglutarate. Alpha-ketoglutarate comes out and goes back to glutamic acid, and so we see a couple of these circles going back and forth. That's not really important for this. The important thing is that malate is the shuttle molecule. Okay? And the other important thing is that NADH on the outside becomes NADH on the inside, and this is a very efficient system. Other questions? This happens to people. Yep, happening in you right now. Okay, so that finishes up what I want to say about electron transport and oxidative phosphorylation and respiratory control, which is what that all was really about. Let's talk, we're going to talk about this for the next couple of days, about lipid metabolism. So lipids are what used to be considered the most boring aspect of biochemistry, all right? They're not inspired molecules. However, lipids contain the most energy of any molecules in our body. They contain the most energy. If we compare the amount of energy in a fatty acid to the amount of energy in a glucose, and we allow for the difference in the number of carbons, so there's 16 carbons, for example, in palmitic acid, and there's 6 carbons in a glucose. If we even allow for that, we find fatty acids contain more energy per carbon than glucose does. Okay? If we count up all the different carbons, we count up all the number of ATPs. By the way, an estimate that I will use in this class for the number of ATPs is roughly 3 for each NADH. There's enough protons pumped to make approximately 3 ATPs for each NADH. And people do entire PhD theses on whether it's 3 or it's 2.6 or it's 2.5, and it doesn't really matter. Okay? So roughly 3 ATPs per each NADH, and roughly 2 ATPs for each FADH2. Okay? So if we use those numbers, all of the ATPs produced by the complete oxidation of glucose, okay, taking everything through the electron transport and citric acid, electron transport and oxidative phosphorylation, citric acid cycle, and all of that, all right, we end up with 38 ATPs per glucose. Now that 38, that contrasts with fermentation. Fermentation, if we used all of the energy from glycolysis, we ended up with 2 ATPs because we made lactate. We reduced that NADH to make lactate. We lost the NADHs of that, right? So 38 ATPs is a lot more ATPs per glucose than 2. We're going to see when we oxidize fatty acids for something like palmitic acid, we get about 134, depends again how you count it, 134 ATPs per palmitic acid. Palmitic acid has 16 carbons, but even when we allow for that, there's still more per carbon in palmitic acid than there is in glucose. So lipids are important. We store our energy in fat. Okay? Fat is problematic, though, because fat is water insoluble. It doesn't move in our bloodstream readily. We have to package it up. Our liver has to actually package it up so that it can travel in the aqueous environment of our bloodstream. If we don't package it up, then we get these insoluble globs that clog everything up, and we got a real problem. So I'm sure most of you have heard of LDLs and HDLs, and you hear about them associated with cholesterol. We'll talk about that in a bit, okay? But LDLs and HDLs are ways of moving lipids through the body. They're ways of moving lipids through the body. Packaging lipids up takes time. It takes energy. It takes effort. The body doesn't want to package lipids up unless it has to. It's also not, because it takes a while, it's not a rapid source of energy. Whereas glucose is water soluble. Glucose can be released from the liver into the bloodstream very quickly. We saw the Cori cycle. Okay? So glucose is great for quick energy. Okay? It's great for traveling in the bloodstream very easily, but lipids contain more energy per carbon than glucose do. So we use lipids for longer-term storage of energy. To give you an idea about when I say longer-term, what does that mean? If I put you on a starvation diet where I didn't feed you for a whole day, you would burn up, or you could burn up most of your glucose reserves in that one day. Okay? Now, people eat more frequently than one day. And if you don't eat for one day, or you go on a starvation diet, you don't die. Okay? But you have to find other ways of making glucose. And as I said, we don't have the glyoxylate cycle, so we can't use fatty acids to make glucose. If you're on a starvation diet, the only way that you make glucose is by breaking down protein. Because the amino acids can be converted into intermediates. You've seen, for example, that aspartic acid can be made into oxaloacetate. And oxaloacetate can be used to make glucose, right? So your only source of glucose is protein. Okay? Your only source of glucose is protein. When you're on a starvation diet, get that right. Okay. When you're on a starvation diet, the only source of glucose is protein. Now your body needs glucose. Your eyes need glucose to function. Your brain needs glucose to function. When you don't have glucose, you get hypoglycemia. And this is when people faint, okay, from too low of a blood glucose. It can be life-threatening in some cases. So you've got to be careful with that. Diabetics in particular will have some problems with managing blood glucose, okay, because that normal balance that's there is not present. And so it has to be managed very carefully. Okay. Let's think about the fat metabolism. Okay. Here is a fat, also known as a triacylglycerol. Fatty acid, fatty acid, fatty acid, attached to a glycerol. Dot, dot. Triacyl, meaning 3 fatty acids on a glycerol, triacylglycerol. If I use enzymes called lipases, I can cleave those fatty acids off, okay, using water. They're hydrolysis reactions, and I'm left with glycerol and fatty acids. I can also get fatty acids off by cleaving them with cleaving glycerophospholipids with phospholipases. So 2 ways I can get these fatty acids. One is by cleaving triacylglycerols with lipases, or by cleaving phospholipids with phospholipases. Phospholipids are the same thing as glycerophospholipids, right? They both give me fatty acids, okay? So it's the fatty acids that we're going to focus on with respect to our next metabolic processes. Yes, John? That is a fat. Yep. By the way, fat is not stored in our cells. They're stored in specialized cells. The specialized cells that store fat are called adipocytes. A-D-I-P-O-C-Y-T-E-S. Adipocytes are fat-storing cells. We don't store them in our regular cells. Okay, well, let's look at a phospholipase. You've actually seen a little bit of this before when I talked about prostaglandins, but I'll tell you more here. We see here that this is a glycerophospholipid, meaning it has a glycerol. There's the 3 carbons of the glycerol. There's the phospho, and there's the acyl group, glycerophospholipids. Okay, so these guys make it the lipid part because they're the fatty acids. glycerophospholipids, right? Different phospholipases cleave this in different places. And no, you're not going to have to memorize this. Okay? You should know that phospholipases can cleave off fatty acids. They can cleave them off here. Or there are some lipases that will actually cleave the phosphate off as well. Okay? Anybody remember what phospholipase A2 was important for? Anybody remember what it cleaved off of a glycerophospholipid? It cleaves this, but there was a molecule that has a name that we talked about in class that was important, that phospholipase A2 cleaved off. I'll tell you, arachidonic acid. So arachidonic acid is cleaved off by phospholipase A2. That's how we make prostaglandins. Remember the bee sting? The bee sting actually activates this phospholipase right here. You don't need to know that. I'm just telling you that now. Okay. Now, fats, interestingly, can get broken down by a pathway that's controlled by hormones. Fats can get broken down in a pathway that's controlled by hormones. Now, we haven't talked about hormone signaling here in any detail, so I'm going to lead you through this pathway because it's the first time that you will have seen this. This is a cell. This is an adipocyte, meaning it's storing fat. Okay? It's storing fat. There's fat molecule right there, triacylglycerol. In order to break down triacylglycerol, I have to have a lipase to do that. And as we can see in this figure, there are three different lipases that actually act on triacylglycerol. The first of those has to be activated by a hormone-activated process. The first one is the only one whose name I'm going to ask you to know. It's known as triacylglycerol lipase. And people frequently put the words hormone sensitive next to it, but if you just know triacylglycerol lipase and that it is hormone sensitive, that's fine. Triacylglycerol lipase. Now you should remember this enzyme name, and you should remember that it is responsive to hormones. Hormone action activates this enzyme. Let's see how this hormone does this. Okay? There are hormones like adrenaline that will activate or interact with certain proteins on the cell surface. Okay? Certain proteins on the cell surface. They're called, well, I'll give you the name. I won't hold you responsible for it here. The name is called the beta-adrenergic receptor. Okay? It's just a protein on the cell surface. When a hormone like epinephrine binds to that, it activates that receptor. What does activating the receptor mean? It means that it causes the receptor, okay, to actually, it's a little bit more complicated, but in short terms, to stimulate the synthesis of a molecule of cyclic AMP. Cyclic AMP. You've seen that before. Okay? So the action of the hormone stimulates the receptor to stimulate the production of cyclic AMP. What does cyclic AMP do? Cyclic AMP interacts with a protein to stimulate it. This protein is an enzyme, and it's a kinase. It's called protein kinase. So when the hormone binds, cyclic AMP is made. Cyclic AMP activates protein kinase. And what does protein kinase do? It phosphorylates our friend, triacylglycerol lipase. You say, what a convoluted pathway. Well, it turns out that this is a process called signaling. Signaling processes in cells are extremely complicated. I've actually simplified this one to make it more understandable. They usually go through a series of step. There might be 20 steps in a signaling pathway, the end product of which is frequently either the activation or the inactivation of an enzyme. Well, this particular enzyme is important because when this enzyme is active, it cleaves off one of those 3 fatty acids. It cleaves off one of the 3. That's why you see this fatty acid over here. And you see that there's 2 left attached. Another lipase cleaves another one off. Another lipase cleaves another one off. I don't expect you to know these names. I do expect you to know this name. Now, this whole process happens because this hormone bound and made this process occur. If this hormone doesn't bind, this thing sits here in the inactive state and nothing happens. Okay? Make sense? Questions? Yes? Protein kinase phosphorylates triacylglycerol lipase, and that causes it to be active. And yes, there has to be ways of other things to inactivate it. We're not going to focus on that here. We just want to focus on the fact that we have to activate that triacylglycerol lipase to cleave a fatty acid off to start the process. If we don't cleave the first one off, then this enzyme can't work because it only works on ones that have 2. And this one only works on ones that have 1. All right. So we've got a way of cleaving fatty acids off. This fat cell can now export these out into the bloodstream where they can be carried by a protein called serum albumin. Yes? Fatty acids. The fat cell exports fatty acids. Those freed fatty acids can be carried in the bloodstream by a protein known as serum albumin. Serum albumin is the most abundant protein in your bloodstream. That tells you it's kind of important. It carries a lot of things. Yes? Serum albumin? Yeah. Serum, it's 2 words, S-E-R-U-M. That's the serum part. Albumin, A-L-B-U-M-I-N. Serum albumin. So the fat cell is starting to feed the other cells. But what happens to that fatty acid? The serum albumin binds to another cell. Another cell says, oh, look, fatty acid's going to gobble it up, going to transport it into myself, and it does that. So the fatty acid gets inside the cell of the target cell that's hungry. It takes that fatty acid in, okay, and it goes through a convoluted process to basically put a CoA onto it. Remember we had acetyl-CoA? Here we have what's called an acyl-CoA, A-C-Y-L. Acyl-CoA means fatty acid linked to CoA. An acyl-CoA. Notice this is an energy-requiring process, and notice the product is AMP, meaning we've done the equivalent of used 2 ATPs to put this guy on here. Why did we put that on there? Why? Now let's think about this. We had this fat cell. This fat cell released this fatty acid into the bloodstream, and serum albumin gobbled it up immediately. And then when it got to the target cell gobbled this fatty acid up, and it put a CoA onto it. What do you think that the cell is afraid of having free fatty acids there for? Any thoughts? Because that's what it's saying. It's saying, I don't want it in the body as a free fatty acid, and I don't want it in my cell as a free fatty acid. Why do you suppose the cell in the body would be concerned about that? Andrew? That's essentially the answer. Okay? This guy, Tessa? No, the oxygen is not really reactive. Andrew, you get a point of extra credit for this, okay? Because this guy right here is a detergent. And what do detergents do to proteins? Denature them, right? And if they denature them, that's going to be bad for the cell. It's going to be bad for the body. So if you gobble them up and you protect them by putting something like this on them, or by gobbling them up with serum albumin, you stop them from acting like a detergent. Now this also has the purpose, the CoA also has the purpose of being a really useful handle. With this thing on there, the cell knows that this guy over here is useful. That's part of the reason we had acetyl-CoA. That acetyl group was useful in the citric acid cycle. That CoA handle was like an identity tag. This will be useful for you to do stuff to. Yes, Mariah. That is this.
Right, but is that like the fatty acid, or is that?
This is the fatty acid. That's right.
Okay, so the fatty acid acts as a detergent.
Fatty acid will act as a detergent. That's correct. That's how we make detergents. You've heard of saponification? In saponification, what are you doing? You're taking fat, mixing it with sodium hydroxide, so that you pull the fatty acids off of the fat. They combine with sodium to make a sodium salt of a fatty acid. Okay. Now, the cell's gone to a lot of trouble to take that fatty acid and put it onto a CoA. What it does next is going to surprise you. It's kind of like, it sounds like something on the internet. What happens next is going to surprise you. Click here, right? I'm clickbait, all right? This is clickbait. This is going to surprise you guys, right? All right. What's going to surprise you is that wasn't very useful for it. Because that thing with a CoA on it can't cross the mitochondrial inner membrane, and that's where the fatty acids got to get oxidized. So the cell just blew 2 ATP to put a CoA on there that in the next step it's going to take off. And it's going to take it off in the intermembrane space between the outer membrane and the inner membrane of the mitochondrion. Now this is important because fatty acid oxidation occurs in the matrix of the mitochondrion, just like the citric acid cycle occurs in the matrix of the mitochondrion. That means the fatty acid's got to get from the cytoplasm up here into the matrix down here. Outer membrane, inner membrane. Well, fortunately, this guy can travel through the outer membrane pretty readily. Remember I said the outer membrane is fairly permeable. But it gets to the inner membrane, and there's no transport system to move the acyl-CoA across. What's a cell to do? Well, the cell has a transport system that involves a molecule of carnitine. How does it do this? Well, there's an enzyme called carnitine acyl transferase. You can also call it acyl carnitine transferase, either way. All right? That cleaves off the CoA and replaces it with a molecule of carnitine. That now makes an acyl carnitine. We started with an acyl CoA. We end up with an acyl carnitine. The inner mitochondrial membrane has a transport protein that will take that across. It's an odd system? You betcha. Does it do it? You betcha. And then once it gets it in the matrix, the reverse reaction happens. The carnitine is taken off and replaced by a CoA. Actually, right here, CoA. Okay. Now, this guy right here, the acyl CoA, is in the matrix, and it's ready to be oxidized. Questions? John?
So the CoA is put back on?
The CoA is put back on a different CoA. There's a CoA that's floating around in here, but yes. Okay? How did the CoA get there in the first place? These are made inside the mitochondrion. So that's, yeah. So they have to be made in the mitochondrion. They also have to be made outside the mitochondrion. So the mitochondrion, remember, has a lot of different enzymes in it. And it has enzymes for making carnitine and also enzymes for making CoA. Okay? Andrew?
This second
Okay, good question. Does this enzyme also require 2 ATPs? No, it does not. Okay? Because you notice we didn't require any ATPs to put the carnitine on, meaning the carnitine has roughly the same energy as the CoA does, and it's not necessary here. It's a good question, but it's not necessary to add other ATPs. So this is a protein that is specific for that, yes. There's a transport protein in the inner mitochondrial membrane that moves the acylcarnitine, but not the acyl-CoA in. That's why the carnitine's got to get put on. Okay? All right, so now we've got an acyl-CoA that's in the mitochondrion, and we're actually ready to oxidize it. So let's oxidize it, okay? Start up here at the very top. Here's a fatty acyl-CoA, or just an acyl-CoA. You can call it either one. And what we're going to see is that we're going to do a process called beta oxidation. The oxidation of fatty acids is known as beta oxidation. Now, the beta oxidation gets its name from the fact that the first carbon away from the carbonyl is called the alpha. The second carbon is called the beta. I don't use that designation, but you're welcome to. I call this carbon number 1, 2, and 3. Very simple. And what we're going to see in fatty acid oxidation and also in fatty acid synthesis is that all of the action occurs between carbons 2 and 3. All of the action occurs between carbons 2 and 3, also known as the alpha and beta. Okay? There's 4 relatively simple steps, and you've already learned 3 of them, as you will see. What are the 4 steps? The first step is a dehydrogenation. That's different from a dehydration. A dehydrogenation means removal of hydrogens, whereas dehydration means removal of water. So the first step is a dehydrogenation, and that creates a double bond, a trans double bond. Where have you seen that before? No, not quite. That would be dehydrogenating oil. It makes an unsaturated fatty acid, but that's not it either. You've actually seen this reaction once before. The last time you made FADH2, correct, and what was the reaction? There's a point of extra credit. It's a pathway we just talked about recently. Succinate to fumarate is the reaction. In the citric acid cycle, the succinate dehydrogenase reaction, I told you we're going to see some reactions that will appear again in fatty acid oxidation. It's the exact same reaction. If you look on that, between carbons 2 and 3 on succinate, we pull out 2 hydrogens and 2 electrons, and we create something that has a trans double bond known as fumarate. Well, this isn't fumarate because it's got a whole bunch of other carbons on there, but the reaction is the same. And the product of that is FADH2. Exactly the same as the succinate dehydrogenase reaction. Now, this is one of 2 enzymes in this pathway I'm going to require you to know, and you'll see why in a second. This enzyme has a fairly long name. It's called acyl-CoA. That's ACYL-CoA dehydrogenase. Acyl-CoA dehydrogenase. Why do I want you to learn that enzyme? Well, it turns out this enzyme comes in 3 different forms. One form works on really long fatty acids. Very long. Something 22 carbons or more. That's the long chain acyl-CoA dehydrogenase. That enzyme turns out to be found in a different cellular structure called the peroxisome. Found in the peroxisome. We'll talk about that a little bit later. There's a second one that you might suspect if we have one that works on long, we must have one that works on medium chain fatty acids, and we do. That one's found in the mitochondrial matrix, which is where most of beta oxidation occurs. A third one works primarily on short. So medium chain goes from about 16 down to about 8 or so carbons, and short goes from 8 down to 2. Why am I going through all this? Well, it turns out that when we look at sudden infant death syndrome, okay, there's a relationship between low levels of the medium chain one and sudden infant death syndrome. It's not a cause and effect, but there's a relationship that appears to be between the two. Many infants who die of sudden infant death syndrome are very deficient in the medium chain fatty acids. They're unable to get energy from those medium chain fatty acids. So that's an acyl-CoA dehydrogenase. John?
They were low in the medium chain fatty acid or
Yes.
medium fatty acid enzyme?
Medium chain fatty acid enzyme. Thank you for making me clarify that. Okay? They're deficient not in the medium chain fatty acid itself, but in the enzyme that works on medium chain fatty acids. So consequently, they're unable to metabolize those medium chain fatty acids. All right. We made a trans double bond. If you go back and you look at the citric acid cycle, you'll see the next thing that involves an enzyme called fumarase that adds water, right? And when we add water, what happens? Well, in the citric acid cycle, we make fumarate, which has a hydroxyl on carbon number 3 and a hydrogen on carbon number 2, just like this. All the action you can see is, again, between carbons 2 and 3. This is an asymmetric carbon right here. We call this in the L configuration. Okay? Fumarate, when we add water to it, creates L-malate. Right? So again, we're making the same configuration. L, in this case, don't worry about the name. You're not going to have to know the name. You don't need to know this name either. In fact, there's no names here that you need to know except for the enzymes that I tell you. But you should know what's happening in each reaction. Dehydrogenation to make a double bond. Trans, right? Adding of water to make an L-configured compound. Okay? Fumarase catalyzed the reaction in the citric acid cycle that's identical to this reaction. Fumarase is not in here. I haven't told you the name of this enzyme, and you're not responsible for the name of this enzyme. Okay? All right. In the citric acid cycle, malate gets oxidized to oxaloacetate. The hydroxyl gets oxidized to a ketone. Bang, bang. Exactly the same as the citric acid cycle. All the action, again, between carbons 2 and 3. NAD is converted to NADH. So far, we've gotten an FADH2, and we've gotten an NADH. Next step, we're going to break off 2 carbons. We break off 2 carbons using an enzyme that you need to know the name of. It's called thiolase, Thiolase. Thiolase. Clips off 2 carbons, making an acetyl-CoA. Where's that going to be used at? Where does the cell use acetyl-CoA? Citric acid cycle, right? So this is going to go be oxidized in the citric acid cycle. And this guy lacks 2 carbons, and it's going to go right back up here and start again. So this is a cyclic process. Each time it turns, 1 acetyl-CoA is released, 1 FADH2 is produced, and 1 NADH is produced. You've just learned beta oxidation. 2 enzymes to remember: acyl-CoA dehydrogenase and thiolase, the first and the last enzyme of this process. Now, I'll tell you later why I want you to know thiolase, but we'll see thiolase will pop up in another metabolic pathway. Fats, yeah. The fatty acyl-CoA goes right back up here. Now it just simply lacks 2 carbons from what it started with. Yep. All right, we've accomplished a lot there. I think this is a day worthy of 2 songs. One we will sing and one we will listen to. The first one we will listen to. And let me make sure I've got the audio turned on. Okay. Okay. That's one to watch. Let's sing one. And the one to sing, this is a real easy one to sing. It's about fatty acid oxidation. It's called When Acids Get Oxidized. The fatty acids carried by CoA are oxidized inside the mitochondria. They get to there, as you have seen, by hitching rides on carnitine. Then it goes away when acids get oxidized. Electrons move through membranes. Yes, it's true. It's true. They jump onto a CoQ. The action can be quite intense when building proton gradients, and it's good for you when acids get oxidized. The protons pass through complex 5, you see. They do this to make lots of ATP. The mechanism you should know goes through the stages LTO, so there's energy when acids get oxidized. Okay, guys, see you on Friday.